All right, thank you, Elvis, for that kind introduction, and welcome to today's Frontiers in Genetics webinar series. Let's get right into it. So today, I'll be discussing genomic medicine as a discipline that involves using genomic information as part of clinical care, with implications on both diagnostics and therapeutics. And to understand the importance of obtaining a genetic diagnosis through case examples, and also to learn and review the cytogenetics and molecular genetic laboratory tests that are available at genetic test sites. So this is the outline, which I will go over some of the background and the techniques in cyto and molecular genetics. This is not a comprehensive list. However, I do try to cover the most commonly used tests. And finally, we can talk about some case examples to illustrate these tools. So as you already know, the DNA is housed in the nucleus of almost all of our cells. And this DNA is organized into chromosomes and chromatin. And when you uncoil these chromatin, you can then get these double-stranded DNA, which encodes our genetic sequence. And there's 3 billion nucleotides, base pairs in each human cell. And how this genetic information influences the observable phenotype is thought to be through amplification of information from the genetic code into mRNA and protein, its regulation, as well as the integration of these biological molecules will then form genetic networks and pathways that influence cellular function, and they can be organized into organ systems that will have an effect on physiology, ultimately behavior, and the whole organism. There are actually thousands of variants from individual to individual. Most of these genetic variants are not disease-causing. They may simply play a role in certain traits, for example, hair color, ethnicity. But today we'll be focused on mutations that are going to cause genetic conditions. So the scale of genomic variants varies from those that affect single nucleotides all the way to those that affect hundreds and thousands of nucleotides to entire chromosomes. And we know typically even single nucleotides at the important genes can also lead to very severe consequences. For example, a single nucleotide change can cause Noonan syndrome. And copy number variants are those that affect several thousands to several million base pairs. And typically we would think of DiGeorge syndrome or 22q11.2 deletion. And for aneuploidies, we typically would think about trisomy 21 or Down syndrome. And there are various different methods that we use in the diagnostic laboratory to diagnose these molecular pathologies. So here are some of the more commonly used methods on the top. And the variant types that these tests can interrogate. This is a pretty busy table, and I don't expect you to memorize or remember all of these, but we'll go through some of these assays and talk about the variant types that they can detect. So what are the reasons for getting genetic testing? So the first would probably be to diagnose a patient who is affected with a genetic condition. We can also do carrier testing, for example, to determine whether an individual carries a pathogenic mutation that could be passed on to future offspring. We can also do pre-symptomatic testing, for example, testing for later onset diseases like Huntington that runs in the family. There are also other uses, for example, susceptibility, later onset diseases, and even how our body reacts to certain drugs, metabolisms. And having a genetic diagnosis can inform not only the clinical management, it enables a better understanding of the condition as well. It can inform prognosis. And so we use the term genomic medicine a lot these days. It is discipline of using the patient's individual genomic information in clinical decision making. So it demonstrates impact of direct clinical implementation on health outcomes or behaviors. And with the advances in molecular technology, it has led to an increase in diagnostic rates across all areas of clinical genetics, which enables more accurate and precise diagnoses and performs more specific risk assessments. With that, we also need to cover more bases in terms of genetic counseling. We have to ensure that the patients are aware of what tests they are getting, and they understand the possible outcomes of the testing, because not all patients are well versed in genetics. So it takes a lot of time to explain to them what are the fundamental concepts of genetics and inheritance. So there are also the chance of identifying secondary findings. So with this, I'm talking about the ACMG secondary finding list of potentially actionable, medically actionable findings. There can also be incidental findings, for example, mutations in genes that are associated with a disease not related to the referral indication. There can also be unexpected consanguinity that can be picked up by genetic testing, as well misattributed maternity and paternity. And after testing, post-test counseling will also disclose the results to the patient. And sometimes cascade testing would be recommended for family members. So now I'll move to talk about the various different tests that are available at genetic diagnostic laboratories. First, let's go over some of the common cytogenetic tests. So G-banded chromosome analysis, also known as karyotyping, is a method to observe chromosomes under the microscope. So this requires live cells because we have to culture the cells and in a controlled manner to arrest them at metaphase. And we want to arrest them at metaphase. It's because this is when the chromatin, the DNA, is condensed into chromosomes, which we can then tell the banding. And so when the cells are arrested in metaphase, we will add certain solutions to swell the cells and also burst the red cells. And we typically use hypotonic solution. And we would fix these cells and then drop them onto a glass slide. And the dropping action will actually burst these cells, and the chromosomes would spread onto the slide. And after that, we will use Giemsa staining to band the chromosomes so we have recognizable patterns to observe on the microscope. And we will identify numerical abnormalities, so abnormal chromosome numbers. We'll also identify deletions and duplications across the genome. And we can also identify rearrangements such as translocations, inversions, insertions, different abnormal chromosomes like ring and marker chromosomes, and so on. So this is a metaphase spread. Cytogeneticists would be able to analyze the chromosomes in this form, and you can already tell the abnormalities if there are any translocations or inversions observable. And we can also cut these chromosomes out by a computer software and line them up from 1 to 22, as well as XY. And then this way, we can compare band to investigate any gains or losses or any smaller abnormalities. So the resolution can vary from preparation to preparation, as well as across different cell types. So typically, we would aim to analyze blood chromosomes at 550 band level. And for tissues, they're typically a bit shorter at 300 to 400. This refers to the number of bands that you can see across the genome. So each band is approximately 3 to 7 megabase. That's why the detection limit of gains and losses is typically quoted at about 5 to 10 megabases. So this is a a sample of peripheral blood, you can see many bands. The chromosomes are very long, very stretched out, and you can clearly see a lot of detail. In the middle, we have amnio and CVS. Typically, these are a bit shorter. You see less bands. And this on the right side is marrow. And these are a lot shorter, and there aren't as many bands you can observe. And for the nomenclature, we would refer to the ISCN 2020, which lists out the proper way of annotating cytogenetic findings. So if you're interested, you could take a look at ISCN 2020. There's also going to be an update that would be coming soon this year or next year. So karyotyping is useful for the investigation of individuals with multiple congenital anomalies. They are also useful for disorders of sex differentiation. If the family has a chromosome abnormality that is known, we can also use chromosome analysis to investigate different family members. It's also useful for infertility to look for rearrangements such as balance translocations, miscarriage as well. There may be aneuploidies that we can detect. Donor screening. So if people are donating, for example, marrow or sperm or oocytes, they can also just check the correct chromosome complement before doing it. And then also oncology, we can detect a lot of different structural abnormalities that drive certain hematological malignancies. So the advantage of karyotyping is that it gives you single cell resolution. You're looking at individual cells, so you can tell different clones from another. Next, we'll talk about FISH, fluorescence in situ hybridization. So this is a approach where we label a target DNA with fluorescence, and we hybridize that onto the patient's DNA. And through a microscope, we can then count how many signals there are in the patient cell. And this can identify specific chromosome regions. So we need to know the target. It's not a genome-wide approach. We have to have targets. And we can tell deletions, duplications, or rearrangements as well. There are many applications for FISH. First would be rapid prenatal diagnosis. So you can use centromere-specific probes to rapidly detect trisomy 13, 18, 21, and the sex chromosomes. It can also be used for DSDs as well. And if you find copy number gain or loss from another methodology, you can also target that specific region to confirm by FISH. You can also, for example, target the DeGeorge region or the NF1 region as examples. And also, we typically would use FISH to investigate structural rearrangements in cancer. So certain common fusions, we can target those fusion genes to perform cancer diagnosis and subtyping. So here are some examples of FISH. On the left side, this is metaphase FISH. So these probes are hybridized, and then we can tell which chromosome they're on because these are chromosomes in metaphase. And on the right side, this is an interphase cell. You can see the chromatin is organized into chromosomes, so we can count the signals, but we won't be able to tell you where in the genome they are, unlike the metaphase setting. Okay, so next we'll talk about CMA. The microarray refers to a chip that has multiple arrays of sequences in which we know the genomic location of the patient. So by mixing the reference sample with a patient sample and then quantitating the balance between the two, for the reference, we would use a normal reference. And by looking at the fluorescent signal, we can then tell in certain regions of the genome, there are gains or losses in the patient sample. So there are two major platforms, one being the array comparative genomic hybridization, which uses the comparing aspect, which is to mix a normal control sample with the patient sample. And the other platform is a SNP array. The SNP array typically will not require you to mix reference sample with it. You can just hybridize your patient's DNA onto the SNP array, and that would be able to tell you gains or losses already. So the reason for using CMA is that it greatly improves the resolution for the detection of copy number variants, so gains and losses throughout the genome. So for this example, there is a very small 4 megabase deletion in 1p36. For the chromosome analysis, even though you have a very long chromosome with many bands, it might be quite difficult to detect a very small deletion, especially when it doesn't involve more than one band, or if the resolution is not well. But on the CMA, you can clearly see a large segment with a signal consistent with deletion. And comparing the three different technologies, for karyotype, the typical limit of detection is about 5 to 10 megabase. We can see the entire genome with single cell resolution. FISH, we can have a higher resolution, but this is targeted, about 20 to 50 kb per probe. And this is also at single cell resolution. For CMA, we have enhanced resolution of CMV detection across the entire genome. However, we do lose that single cell resolution because this is DNA extracted from cells. So all those signals are pooled into one. But we would have a higher resolution for CMV detection. And the clinical applications of CMA can include the investigation of individuals with multiple congenital anomalies, intellectual disability, developmental delay, oncology, and more. And with the implementation of CMA into clinical practice, we have recognized many recurrent microdeletion duplication syndromes across the genome, many of which are mediated by low copy repeats and non-allelic NAHR, non-allelic homologous recombination. So next we'll move to molecular genetic tests. First, I'll talk about quantitative fluorescence PCR on short tandem repeats and its applications. So this method is a PCR-based method in which we're trying to amplify polymorphic short tandem repeats. So different individuals can have different repeat numbers, and by PCRing the region and sizing them through capillary electrophoresis, we can then differentiate different alleles in different samples. So for example, patient A has repeat size of 4 and 8. Patient B has a repeat size of 6 and 10. And we can multiplex this into panels to investigate these STR markers across multiple chromosomes. One example would be to use a panel of STR markers located on the common aneuploidies, so 13, 18, 21, and also the sex chromosomes. And by counting how many peaks you see, you can then detect either a gain of the entire chromosome or a loss of those chromosomes. So in normal situations, we have 2 copies of each autosome, and you can see 2 peaks like this. When there is a trisomy, you can have a 2-to-1 ratio where you have 2 copies of one repeat and 1 copy of the other allele. You can also have 3 different repeat sizes. So we get a 1-to-1-to-1 ratio, but with 3 different alleles. However, when the repeat size is the same across the 2 disomic alleles, then it would be uninformative because you just see 1 large peak. Okay, so the applications of QF-PCR include the detection of common aneuploidies. This is typically very fast. We can get results in about 5 to 6 hours. Also, the sex chromosome aneuploidies. We can also use it to rule out maternal cell contamination in prenatal samples. So if the sample is blood stained, typically there is a chance that there is maternal sample inside amniotic fluid. So we would always do QF-PCR to rule out MCC before testing it to ensure that we're not testing the maternal sample alone. We can also use it to confirm by parental inheritance of chromosomes. In certain chromosomes, there can be issues when both of the patient's chromosomes are inherited from one parent, for example, UPD15. We can also use these STR markers to perform phasing of genetic variants. This is also useful in pre-implantation genetic testing. And we can also use it to test biological relationship. And similarly, it can also act as DNA fingerprinting in forensics. So now we'll move on to Sanger sequencing. So Sanger sequencing is a targeted approach to sequence a short piece of DNA in the genome. So to begin, you have your template DNA. You would add the different DD NTPs. So these are dideoxy nucleotides. When these are incorporated into the PCR reaction, it will terminate. So no other bases can be built after it. There's also the polymerase, the naturally occurring NTPs, and then the primers. So with this set of reagents, you can then amplify the target DNA into a ladder of different terminating PCR products. And these are all going to differ in size of one nucleotide, right? And when you run this through a capillary electrophoresis and investigating the fluorescence signal that is given off as each of these products pass through, you can then give out a sequence of the nucleotides of your original piece of DNA. So this is especially useful for detecting sequence variants such as SNVs, and it can also be useful for the detection of small insertions and deletions. So this is a fast and cost-effective way to read the sequence of a small region of the genome. It is useful for example, testing of known familial variants or confirmation of variants that are picked up by other approaches such as NGS-based methods. Now we'll move on to NGS. This is a very high throughput method which enables the sequencing of entire human genomes or exomes. The workflow is to prepare the DNA for sequencing, which is called library preparation. Typically, this genomic DNA would be sheared and adapters would be added to the sides, which enable them to bind onto your flow cells for sequencing. So I'll talk about more of the workflow in the subsequent slides. It can be performed using a targeted approach. For example, exome sequencing, we're capturing only protein coding regions for sequencing. This can be done by either hybridization method or an amplicon-based method. If we're doing whole genome sequencing, then we do not need to enrich or capture the segments. So as I mentioned, library preparation methods typically require shearing of the DNA and adding the adapters onto the ends of the DNA. Doing QC to quantitate the library concentration, and then we can equally multiplex multiple samples for sequencing. In terms of the capture methods, we can use amplicon enrichment, meaning we are adding specific PCR primers to amplify only target regions for library preparation. On the other side, we can also use hybridization method, which we will prepare the library, and then we will use a bead-based method to capture the target sequence and only sequence that for sequencing. So the sequencing chemistry varies between different platforms. So for this specific one, I'm going to share, it entails the DNA library being hybridized onto your flow cell, and this single piece of DNA will replicate itself to create a clone of identical DNA, and this is called a cluster. So this cluster would then undergo sequencing base by base, and then each base carries a different fluorescence signal. So by capturing an image one by one after each base is built, you can then read out the base calling. And typically millions of these clusters are being sequenced at the same time, which enables the high throughput aspect of NGS. So for variant calling, the NGS reads are aligned back to the human reference genome, and typically we would detect any variants that are different from the reference sequence. And these variants, we would then interrogate it for population frequency and also other databases to annotate their pathogenicity. So there are many, many applications of NGS. We can perform genome sequencing, exome sequencing. We can do low-pass genome sequencing, which is a shallow read depth-based approach. We can do mate pair genome sequencing, which is useful for the detection of structural rearrangements. NGS can also be used for cell-free fetal DNA screening. We can also use it for NGS gene panels. So typically these would be from a few to hundreds of genes, target genes, and we can also perform that on mitochondrial DNA as well. NGS can be used for RNA sequencing and metagenomic sequencing and more. So next, this is exome sequencing, showing you the pileup of reads across the genome. And on the bottom here, these are exons and introns of the ATP7B gene. And you can see the reads typically would pile up on top of each exon. And the reads and the capture will also include about plus or minus 10 base pairs into the introns. So we would typically be able to capture canonical splice site variants as well. And the exons comprise of approximately 2 to 3% of the entire genome. So by sequencing the exome, we can save costs on the sequencing. The advantage is that because we're targeting a small part of the genome, we can sequence it to very high read depths. So typically exome sequencing can be done at about 150 to 200 X coverage. This means each nucleotide here has 150 to 200 reads that support it. And this would give you pretty good variant calling ability, even for mosaic variants. This can be used for the detection of SNV, small insertions and deletions, CMV detection ability as well, but it might be limited. This is because inherently some exons might not have as many reads supporting it. It could be due to GC percentage. It can also be just alignment, or it can also be due to capture-based factors that can make this read depth vary from exon to exon. For homozygous deletions, it can be quite clear, but for heterozygous deletions and duplications, it might be challenging. And typically, multi-exon events are easier to detect than single exon events. So this is the same view, but for genome sequencing. So you can see both exons and introns have pretty even coverage, and the coverage is not as high as that of exome sequencing because we have a bigger target to sequence. So typically, clinical whole genome sequencing is sequenced to a depth of about 30 to 40x. And this can give better ability to detect genome-wide CNVs. We can also detect SNVs, small insertions and deletions. However, with lower depth, it might not have the same ability to detect low-level mosaic variants. Okay, now we move on to low-pass whole genome sequencing. So as I mentioned, NGS is quite versatile, and we can adjust its read depth to tailor it to our scope of detection and the clinical application. So by doing high read depth genome sequencing, we can detect a comprehensive list of variant types. However, that is expensive and it requires a lot of sequencing power. But if we want to just detect cytogenetic abnormalities, for example, CNVs aneuploidies, structural variants, we can actually sequence the genome at a lower read depth. This way, we can increase the throughput, sequencing cost, and the sequencing depth. So for CMV detection, we can actually perform sequencing at 0.25x, which is already sufficient. And for SV detection, we'll just have to aim for a physical coverage of about 8.25x. And this is the low-pass genome sequencing analysis and CMV detection workflow. Basically, the reads would be filtered through QC steps, so the poor quality reads are thrown away, and these higher quality reads are aligned to the genome. These reads fall into bins. We'll calculate the number of reads within each bin, and higher coverage areas we can call as duplications, and lower coverage areas we'll call as deletions. We can also use a modified library preparation method called MAPAIR genome sequencing, which would enable the detection of structural rearrangements, such as translocations and insertions. And this is utilizing large DNA inserts of about 3 to 8 kilobases. And by doing that and sequencing the two ends of a very long piece of DNA, we basically would increase the chance of capturing a breakpoint that lies between the two reads. When we get this sequence and we do alignment, we can see that this piece aligns to chromosome 16 and the other side aligns to chromosome 17. In usual situations, you should not see a piece of DNA that connects 16 to 17, right? So if we see multiple reads like this, then we can use it to support a translocation event that occurs. So for example, the discordant read pairs would lie between these regions here. Okay. Talk about multiplex ligation dependent probe assay, MLPA for short. So this is a molecular method to detect CNVs as well as single nucleotide variants. So the technology uses these probes that hybridize onto your target sequence. And if there is a perfect match, the two probes, the left probe and the right probe, would be ligated, right? And then the PCR reaction basically would amplify your ligated probe. So if your probes are not ligated, your PCR reaction wouldn't go through. And so this is actually PCRing the probes instead of your sample's genomic DNA. And so there's also a stuffer sequence here that is a sequence with a known nucleotide length. And by having this stuffer sequence, we can calculate the fragment size by adding your target with the stuffer. And you can multiplex this basically, typically we would do multi-exon MLPA. And by multiplexing it, we can then investigate the entire gene for CNVs as well as SNVs. Its applications include detection of copy number variants, and also deletions and duplications of genes and even individual exons. It has very high sensitivity to detect single nucleotide variants as well. Even if there's a one base pair mismatch under the probe, it probably will not anneal. And you can also use methylation specific MLPA analysis. So this is an example of MLPA results. Typically, there are three components. The first is the electropherogram. This is the gel capillary results of the different fragments going through from small to large. You can have the reference sample and the patient sample, and then you can compare which peaks are missing or decreased. So over here, you have the copy number plot. It's showing 3 probes with a deletion signal. And these 3 probes correspond to exons 7 and 8 of this gene. So there is a heterozygous deletion of the 2 exons. So here it's showing you the gene from exon 2 all the way to the end. And then it also has several control regions. Next, we'll talk about triplet repeat disorders. So Fragile X, Huntington, SCA. So there's many of these triplet repeat expansion disorders. And they are hard to detect because of their mutation mechanism. Most of them are caused by expansion of these repeats. One of the molecular approaches that is used frequently is triplet repeat prime PCR. Conventionally, we would aim to amplify the triplet repeat with a forward primer and a reverse primer flanking the repeat region. So the risk of just doing this is that you can amplify small repeats. However, when these repeats get very large, basically the PCR fails for the larger allele, and this would result in allele dropout, which would look like homozygous normal in that case. So you can add primers that also prime to the repeats. And this would basically give you random priming throughout the triplet repeat, giving you a ladder approach of different fragment sizes. And we would be able to tell there is a larger allele that cannot be amplified. Basically, you would see this ladder approach indicating that there is a larger allele. This is just another schematic showing an example of FMR1 expansion of the CGG repeat in the 5' UTR. You have a full-length expansion here. If you use the triplet repeat prime PCR, you get a series of different fragment sizes, which would show as a ladder appearing signal on your electropherogram. And there's a very large product that might or might not be amplified, but this ladder signal indicates there is a very large product. In those very large cases, we can not size it correctly using PCR methods. We can then use southern blotting, which is a gel-based method. We can run the expanded products through this gel, and we can hybridize the DNA with our complementary or target sequence. And after that, we can use X-ray to radioactive X-ray to expose it to a film. And with that, we can then tell the size of the different fragments. The limitation of southern blotting is that it has a very long TAT, approximately 1 week. It also requires radioactive use as well. Okay. Next, we'll go through methylation testing. So in general, gene expression is biallelic except for certain chromosomes that are imprinted. For example, chromosome 6, 7, 11, 14, 15, and 20. And in these cases, if you do not have biparental inheritance of these chromosomes, it can result in a clinical phenotype due to imprinting disorders. So typically, these expressions are controlled by methylation. And by testing for methylation, we can then tell whether or not one of the alleles is missing. For example, we'll go and talk about Prader-Willi syndrome and Angelman syndrome. Okay, so this 15q11.2 region is the location of the Prader-Willi region and the Angelman region. And for normal phenotype, you need to have both the paternally expressing chromosome 15 that you get from your father and the maternally expressing chromosome 15, which you get from the mother. And if you lose one or the other, you would get a clinical phenotype. And by doing methylation-specific testing, we can tell whether or not individuals have both of those biparental 15s that are expressing. There are restriction enzymes that are sensitive to methylation in which it will not cut the methylated allele. And by running the digested DNA through a southern blot, you can then tell whether or not an allele is missing. So over here, you can see the methylated allele is not cut, so it's bigger. And the paternal allele, which is unmethylated, is cut, so it's smaller. So in normal situations, you have both alleles. And in Prader-Willi syndrome, you're missing the paternal allele. And in Angelman syndrome, you're missing the maternal allele. And this is just the X-ray film showing the two fragment sizes. There are also other genome-wide methylation assays. For example, EpiSign. This is a methylation array-based method which can detect methylation defects of over 90 conditions. And these are some of the examples of genetic conditions that have a specific epigenetic signature that can be recognized. Here showing Rubinstein-Taybi, Smith-Magenis syndrome, Sotos, Turner, and more. So there's about 90 syndromes that this array can pick up. So next we'll talk about long read sequencing. It's becoming a method that can potentially make its way to the diagnostic laboratories. So by short read sequencing, we are sequencing the entire genome, but there are gaps. And these gaps are caused by complex regions that cannot be well sequenced. For example, repeat regions or high complexity regions. We call these dark areas of the genome. So in the reference genome, GRCH38, these might be gaps that we basically cannot align any reads to. With long read sequencing, your sequencing reads are over 10 to 15 or even longer kilobases. And this will allow you to better represent the genome by sequencing through those complex regions, filling up the gaps and potentially identifying more disease-causing variants. So there are two approaches. One is the nanopore sequencing, and the other is the single molecule real-time sequencing. Right now, it can be used for enabling more precise resolution of complex structural variations, and it can also resolve previously undiagnosed cases. So optical genome mapping is also another emerging method in the clinic. This involves labeling DNA by an enzyme that recognizes a six nucleotide hexamer. So this DLE enzyme would go through the genome and identify regions with CTT-AGG hexamer and put a fluorescence tag onto it. And this CTT-AGG hexamer occurs at about 14 to 17 times every 100 kilobases. So you would get hundreds and thousands of these marks across your chromosome. And by linearizing these very long DNA that are barcoded and imaging them, you can then digitalize these molecules, align it to a reference genome, and you can compare the patient sample with a normal reference sample to detect structural rearrangements. So here is an image showing you these single molecules that are digitalized, aligning and building these longer contiguous maps. And by comparing it with your reference genome, you can detect deletions, duplications, inversions, insertions, translocations, and even repeat expansions. It also has several targeted applications, including the detection of FSHD type 1. So FSHD is a disease that is caused by a repeat contraction on chromosome 4q at the very telomere end. And this has to occur together with a permissive allele called the 4qa. And so the optical genome maps are able to tell you that A, there is a contraction, and B, there is a permissive allele. And when that occurs together, this would be a diagnosis of FSHD1. It can also be used for Fragile X syndrome analysis. So there is a dedicated pipeline that looks at the FMR1 5' UTR to identify the CGG repeat size. And basically, the software will calculate a probability that the expanded allele is exceeding 200 or not. So next, I'll talk about a case example and several studies. So this is a 5-year male with EB simplex, where the parents were asymptomatic. an EB panel was sent, and this only revealed heterozygous VUS in two genes, DST and LAMC2. So these two variants are in genes that are associated with EB simplex. However, there's only a heterozygous variant, and the second hit was not found. So we cannot conclude the diagnostic finding for this individual. When whole genome sequencing was sent, it identified a de novo pathogenic variant in KRT10. And this variant has been reported in several patients with epidermolytic hyperkeratosis. And this is quite consistent with the clinical features. And this was considered a diagnostic finding. And the reason why this was not picked up was because the gene panel, for some reason, did not include this gene. So you have to be aware of the size of the gene panel as well as the gene content when ordering and trying to exclude certain diagnoses. And next, I'll talk about two studies. Through my time at Baylor, I looked at a cohort of over 13,000 pediatric referrals for high-resolution CMA. And these individuals were mostly referred for developmental delay or multiple congenital anomalies. And the array was very high resolution in that it can detect exonic CMVs of over 42,000 genes. And by that, I looked at monogenic CMVs, meaning CMVs that are affecting only single protein coding genes. And pathogenic or causal small CMVs affecting single protein genes were detected in about 10% of cases who receive abnormal findings. So this highlights the importance of their contribution to CMV disease-causing alleles, and they are challenging to detect because of their size. So over here, you can see the 100 molecular findings in 180 individuals. They're spread across different diseases, mostly about half in autosomal dominant diseases, about 20% in X-linked conditions. And then this shows the distribution of their sizes. The median size of a loss is only 28 kilobases, and the gain is a little bit larger at 53 kilobases. So the implications of this is that we are starting to recognize these very small intragenic CNVs that are disease-causing, and they do have implications on our other genomic methodologies. For example, a lot of people are using genome sequencing as well to detect not only SNVs, but also CNVs. And we need to be aware of these small CNVs when performing these other methodologies. And the next is talking about resolving multiple molecular genetic diagnoses by whole genome sequencing. So as you know, genome sequencing is a very powerful, and it can detect a broad spectrum of variant types. And it detects most disease-causing alleles that are handled by different standard of care tests. And so it has an advantage of being unbiased in the variant interrogation. So in about 2,200 pediatric referrals for whole genome sequencing, about 8.6% of those who received abnormal findings actually had more than one molecular diagnosis. So here showing 53 individuals had dual diagnoses, 4 had triple, and 1 even had quadruple diagnoses. And these diagnoses also occur in different disease mode of inheritance. Interestingly, for comparing the AD and AR conditions in single diagnosis and multiple diagnoses, we see sort of a drop in the AD disorders. So this could be due to hypomorphic or not as severely presenting clinical features that can be diagnosed by molecular interrogation, giving rise to these multiple diagnoses. And in many cases, more than one variant type contributed to multiple diagnoses. So on the right in this pie chart, you can see that only a small portion of those multiple diagnoses were contributed by a single variant type. The majority included a combination of SNVs and CNVs, triplet repeats, mitochondrial variants, and these would typically require multiple tests to detect if we're not using whole genome sequencing, which further highlights the advantage of using whole genome sequencing as a test to interrogate various variant types. For patients who present with a complex phenotype that is not immediately recognizable as a certain distinct syndrome, whole genome sequencing may be helpful because you never know whether or not those are overlapping phenotypes of multiple conditions. And also you have a very broad differential, which means a broader interrogation approach may be more helpful. And this also may change the traditional genetic testing paradigm, which traditionally would involve sequential testing or stopping at the point of establishing a single diagnosis, in which we may miss some of the additional diagnoses. So I'll share with you one last case of a triple diagnosis. This is a female born at 40 weeks and 1 day. She had spit ups and vomiting, hypertonia, hyperammonemia. She had to be put on mechanical ventilation and decreased urine output. So whole genome sequencing was sent on day of life 6. And other clinical indications involved situs inversus totalis, dextrocardia, mitral regurgitation, and some heart defects. Hyperammonemia, metabolic acidosis, encephalopathy. There's also hypertonia and respiratory failure. And there's also coagulation abnormalities as well. So WGS revealed 3 diagnoses, the first one being a heterotaxy in the gene PKD1L1. This was a homozygous variant inherited from both the mother and the father. This was classified as pathogenic. The second is arginosuccinic aciduria, a canonical splice site variant in ASL. This was also homozygous, inherited from both the mother and the father, and this was pathogenic. And the third one is a 17q12 recurrent duplication disorder, and this was de novo, and this was also pathogenic. So this individual had 3 molecular diagnoses, and the first being a heterotaxy syndrome, which is consistent with their heterotaxy in the heart. And the second diagnosis was ASL deficiency, and this is a urea cycle disorder. There is a problem with the ASL enzyme resulting in a block and a buildup of arginosuccinic acid. This can be characterized by hyperammonemia, lethargy, risk of hypertension, as well as hepatic fibrosis and cirrhosis. So quite consistent with the features. And lastly, the 17q12 duplication, which is characterized by developmental delay, hypertonia, some of the neurodevelopmental abnormalities. This is also a reduced penetrance locus, and there's variable expressivity. Nonetheless, this was a pathogenic finding. So in conclusion, there is a multitude of genetic tests that are available to interrogate the genome across diverse variant types. And resolving these multiple molecular diagnoses can inform not only management, but prognosis and recurrence risk. And there are new diagnostic applications that are on the horizon with newer technologies such as long-read sequencing and optical genome mapping. So I hope you enjoy my talk, and I would be happy to answer any of your questions.